Well, we've finished the very, again, cursory, but very introductory portion of Chapter 2 in terms of some of the terminology for our chemistry of the human body. And now we're going to do something similar to that when we talk about a hypothetical cell. Many of you, once again, have had this in some capacity in your high school education and middle school education. Those of you who had additional biology or physiology know this in much greater detail. For those of you who this is brand new information, do become familiar with the terms in your text as we start bringing up some new words, some new terms that you possibly have never heard before. It might help you to put together some flashcards for yourself if some of these things like a Golgi apparatus or a lysosome is a brand new word for you. In addition, at the end of your book you have a CD and there are some animations for the topics involving movement of product in and out of the cell, such as diffusion and osmosis. Your animations on your CD are what I'm going to refer you to, as well as the descriptions in the text to review that process. So let's just talk about initially the basic anatomic hypothetical structures. And when we look at this particular cell, this is a hypothetical cell. This is not any particular cell in your body, but we know that each and every one of your cells has a need to have many of these what we call organelles present to enable the cell to function properly. Now, within the center of most all cells, but some cells don't necessarily, by the way, have these. There's a nucleus, and the nucleus of the cell is the control center, kind of like the cell's little brain. It is the largest of all the organelles, and it is composed of DNA, which is the prescription for the behavior and structure and function of that cell. Wrapped around the entire cell is a plasma membrane, and that is the doorway to the inside of the cell. And in your text on figure 3.1, there's a fairly good pictorial of a plasma membrane. It is made of a phospholipid bilayer supported also by cholesterol. And what's really interesting is that all of your plasma membranes have a marker on them. It's actually called an integer protein, and that particular marker represents the DNA that is prescribed in the nucleus. So when we start talking about our immunologic system and our self versus non-self, the plasma membrane not only lets certain nutritional products in and waste products to go out, but it also is a little call-out marker called the integer protein that says, you know what, my name is Dawn, so this is a Dawn cell. If I were to get a cell, say from a donation of either blood or a donor of a tissue type, those cells don't have the same integer protein representing their same exact DNA. So I really only like the ones that are like myself, as do your cells. Self versus non-self is very important when it comes to disease process in a type of disease called autoimmune disease. So when we talk later in the lymphatic system, as well as those of you who take GH126, we're going to talk a lot about self versus non-self and how, for example, if you have a certain substance get into your body that is recognized as foreign, why your immune system might say, hey, there's a little integer protein, there's a little protein marker on that thing and it's not me and I better start attacking it. So this plasma membrane around our cells is essential for those three factors. It's the doorway to the inside. It allows certain product in, certain product out. We're going to talk about the mechanisms of how those products move, and importantly, it marks it as self. Cytoplasm is the fluid within the cell, or the gelatinous-like substance that fills the cell and holds the other organelles in place. It is the liquid part of the cytoplasm is called cytosol. Now we'll talk a little bit more about some of the other organelles within this hypothetical cell. One is called the endoplasmic reticulum, and it's within this particular organelle that protein is sorted and lipids are synthesized. So So that is the location of sorting of protein and the synthesizing of lipids. And that, again, is the endoplasmic reticulum. A very important organelle is the mitochondria. We call this the powerhouse of the cell because it is that center that really generates the energy. It's the energy producing center, and the name of the type of energy that's produced at the end of cellular respiration is something called ATP, or adenosine triphosphate. Without that energy, all cellular processes cannot progress. So we do definitely need this powerhouse of the cell to be functioning properly and receive the things that it needs to be able to make the end product. And that includes, by the way, oxygen and water and sugar. Also within the cell is something called the Golgi apparatus, and this particular organelle makes proteins and prepares them for transport within the cell. Remember that proteins are essential. Oh, sorry about that. I'm going to have to delete this just for a second. I apologize. I'm doing this from home, and I was afraid my little puppy would do his noise, and I apologize, and I'm not sure how to go back on this. So under Golgi apparatus, if you get a big bark, I apologize for that. That was Gus, by the way. He sometimes likes to participate in my recording here. All right. So again, makes proteins, prepares them for transport. Also within the cell is an organelle called a lysosome. The lysosome is that part of the cellular organelle that digests substances that are introduced into the cell. In addition, there's another called a peroxisome, and that particular organelle breaks down harmful substances. When we talk about the organs of the body, it's interesting to note that many of our large organs have similar functions to some of the organelles that we're talking about. So for example, as we learn about the liver in your body, it is a large organ that is responsible to break down harmful substances that we may ingest into our large organism or the human body. Other types of intracellular structures that may be found but not in all cells. One we will find in all is called a centriole. A centriole is as we talk about mitosis and you watch the video, is a anatomic portion of the cell that's important for the splitting and dividing of cells into daughter cells called mitosis. Some cells, actually only one human cell, also might own a structure called a flagella. And the flagella help move cells, again, such as those that are attached to and found on a sperm cell. It is the only human cell. So if I were to ask you, a question in making reference to a human cell that has a flagella, obviously you would not say that we could find one in the female, but we would only find one in the male. We are in the micro class going to talk quite a bit about other types of invaders that have flagella, but in the human cells, the only one again is the sperm cell, and it aids in motility and swimming. Now, as we talk now about that plasma membrane, we know that we have to get product in, product out for, if you were to talk about the organism of the human organism, yes, indeed, the product in would be brought in via the enteric or the mouth, for example, system, part of the digestive system. We know that we can introduce food product in that particular way. Well, think about even eating. Eating takes energy. You need to add energy to masticate or to chew your food. When we talk about moving substances in and out of a cell membrane, sometimes it can passively flow into the cell, and sometimes it can require additional energy to get product in or to get product out. Now, diffusion, osmosis, filtration, active transport, phagocytosis, and pinocytosis will all be explained very well in your animations. The most important thing to note is that diffusion is and osmosis occur by passive transport. Active transport, once again, requires the movement in and out of the cell wall with additional energy. Phagocytosis and pinocytosis, they both require additional energy. Filtration requires pressure. Filtration is not unlike, or the movement of fluid is not unlike when you make, for example, a cup of coffee in a drip coffee pot maker. When you have your coffee pot maker and you fill it with water, and the water gets sucked up by that little tube, and then it drips down under a certain amount of pressure through the ground up coffee bean, that then on the other side of the coffee bean has a filter like, you might have one that's a permanent filter or one that's a cloth filter that is kind of semi-permeable. It only lets certain of the product come through. So hopefully you're not eating coffee grounds in your coffee, because under pressure and using a filter, you're able to get a filtrate, which happens to be the coffee that you drink. Well, our kidneys do a very similar type of thing. When our blood under pressure enters the kidney, it produces a filtrate that eventually, after going through many different processes, becomes your urine. Filtration requires a movement of fluid under pressure through a semi-permeable membrane. Do read about these particular movements of substances through the cell membrane. Watch the animations that are at the end of your CD-ROM for your online class here. If you have questions, please email me. These are very important concepts to acquire, because they relate to many other aspects of both cellular and pulmonary respiration, as well as circulation, as well as, for example, in phagocytosis, protection against invaders. Again, I do want you to take the time to watch the animations, and you can go by chapter to review those. And then if any questions, please either email me or place them on the discussion board. And that will end movement through the cell membrane. The other piece of this particular chapter discusses mitosis. Mitosis is our way of maintaining the bodies that we have. You make new skin cells continuously. You are producing cells, they die, and then you produce new cells. The new cell is called a daughter cell. Attached to the unit module is an animation on mitosis. It's an additional YouTube video. I would have you first read through your chapter and talk about the different phases of mitosis, and then watch the video to see it in motion. Ultimately, those centrioles help pull and separate the material that's within the nucleus apart, leaving behind an ability to duplicate the DNA exactly as it was, so that we have two new daughter cells with the exact replicated DNA from one to the other. So for mitosis, that is how I want you to review this particular topic. And once again, if you have additional questions, please feel free to place them on the discussion board or email me. You are responsible to understand the basics of mitosis and mitotically dividing. It's especially important because of the fact that normal mitosis makes two identical daughter cells, and unfortunately abnormal mitosis is what is the precursor to the development of of potentially cancerous cells, where instead of making a daughter cell that's the normal one, you take a little tweak and a turn and you make an abnormal, or now a cancerous cell, as a result of abnormal mitotic dividing. So with that, I will let you review those topics also, and additional questions, please forward to me.